Good morning. Today our discussion topic is vinegar production. Firstly, the requirement, as we have discussed earlier, that there are certain prerequisites in producing any industrially important compound. There were three prerequisites. First one was the organism. So the organism which are used for the fermentation of acetic acid, that is your vinegar, is the Acetobacter species. Vinegar production, it is one of the most oldest fermentations product that have been used earlier also. It is a very quite old process of vinegar production, and it has derived from the French word vinaigre, meaning sour wine. So in other words, we can also define vinegar as a sour wine, which is generally produced by letting the wine to go sour under the unwanted natural conditions. And there are basically three methods for vinegar production. Firstly, one of the earliest method was the use of wooden vats. The second method that was used for in the earlier production of vinegar was the use of barrels, and this process was known as the Orleans process. The most commonly employed with better designing is the Fringe method. Now the biochemical reactions that occur in the production of vinegar, there are basically two types of biochemical changes, or we can say two types of fermentation occur while in production of vinegar. So first biochemical change is the alcoholic fermentation of carbohydrates, that is utilization of yeast for the fermentation of alcohol by using a yeast, and mainly Saccharomyces species is used. And the second biochemical change that occurs during the fermentation of vinegar is the oxidization of alcohol into acetic acid. So the first biochemical change that occurs is the alcoholic fermentation, or we can say there are two types of fermentation occurs in the production of vinegar. First one is the alcoholic fermentation of carbohydrates, as we have studied earlier. And the second one is the oxidation of the alcohol, or you can say ethanol, to acetic acid. Now there are different various types of vinegar that have been produced by using different materials, and the differences among them, they are primarily because of the material used. So they are basically the different type of vinegars on the basis of material. They can be categorized into different types, and the materials which are used for the alcoholic fermentation, basically the materials used for alcoholic fermentation, they differ the different types of vinegar, and mainly used materials are sugar syrups, all the carbohydrate containing substances, mainly honey, wine, beer, and some fruit juices also. Technically, vinegar is defined as the fermentation derived food product which contains not less than 4 gram of acetic acid as per 100 ml volume. So we can define it as a fermented food product which contain not less than 4 gram of acetic acid as per 100 ml. And one of such types of vinegar, the definition of one type of vinegar, which is the apple simply vinegar or cider vinegar, or this is also known as apple vinegar. So one definition is given by USDA, that is United States Drug Administration, and it has given the definition of apple cider juice, apple cider vinegar, which is produced from apple juice, and they have given the definition as it is a fermented food product which is produced by the acidification of apple juice. And the standards are, as I have told earlier, the standards are that it contains not less than 4 gram of acetic acid per 100 ml of volume. Generally, the alcoholic concentration that is added into the alcoholic broth it varies from 10 to 13%. So, the alcohol concentration that is added into the broth is 10 to 13%. Now, there are different equipments which have been designed for developing the fermentation process for vinegar production, and as I have told earlier that the first one, the earliest most method used was wooden vats. In the wooden vats, they use this. These are certain wooden containers that are placed vertically, and the large surface area of these barrels, they provided the suitable aeration for carrying out the fermentation. And the Acetobacter species, they were the alcoholic broth that was inoculated by it used to get inoculated by air, by the surrounding air. So, the aeration was through the air, the aeration was through the large surface area of the wooden tanks. One of the disadvantages of this process was that it was a quite slow process, and secondly, the fleas production occurred while the incubation. Eels, nematodes, which were known as the eels, nematodes or eels vinegar, that is these are certain, basically they feed on the vinegar mother, and they are generally produced due to the longer incubation period. They grow on the vinegar mother, and due to the longer duration of incubation, they are generally produced on the slime that is being produced by the Acetobacter species. So, because of it is a quite slow process, it was not further used. Now, the second one was the Orleans process, in which wooden barrels were used. So, it is known as the Orleans process because it was developed in Orleans city of France, and in this one the equipment that was used was the vertical vessel. They were used the large vertical vessels for the fermentation of vinegar, and the holes, several holes were provided in the wooden barrels which provided the aeration, and in this also the different microflora of Acetobacter species was inoculated, and the alcoholic broth, the slime used to form on the alcoholic broth. This method was, again it was a quite slow process, and this method, barrels method or the Orleans process, it was used for the table vinegar production, mainly for the table vinegar, and the packing material used to be generally the wooden battles. And the alcohol, it used to feed the alcohol via the feed inlet. Now, the disadvantage of this process was firstly the slow process as of the wooden vats, and the second disadvantage was the evaporation of ethanol, that is all the ethanol that was being produced, it used to get evaporated. So, there high loss of ethanol was observed in this fermentation. So, second disadvantage was the high loss of alcohol due to the evaporation and over oxidation, because when these barrels were kept for longer duration of incubation, so all the alcohol used to get lost due to evaporation and over oxidation by acetic acid bacteria. And one advantage of this method was that it used to produce certain byproducts. The Acetobacter species, apart from the Acetobacter species, certain non-Acetobacter species were also active, which formed the byproducts like lactic acid and propanoic acid, which used to act as the esters, and they would impart the flavor and aroma to the vinegar. So, this was the advantage of this method. Then the last method was the generator process or the Fringe method. Because of its modified design, it is now popularly used for vinegar production. So, Fringe method, it is also known as the trickle method or the quick process. And the German process. And it may also be called as generator process, as it employs the generator. Here also Acetobacter species was the microorganism. Generally, we cannot figure out the Acetobacter species, since the microflora all of the Acetobacter species is used. So, we cannot determine which species has led to the formation of vinegar. One species of Acetobacter species is the Acetobacter Selenium, which is found to be harmful as it produces lime on the alcoholic broth. This method was developed in 19th century by Schusenbach. It was known as the quick process as it was very fast. The fermentation used to occur very fast, and in this one a large vertical tank was used, which was made up of the wooden vats. It was basically of stainless steel, and this material, the supporting material, was of wood. It was either open from the bottom or the top, and the proper aeration inlets were provided in the bottom of the tank. So, the packing material which was used in this method was, they may be the corn cobs or the beechwood shavings or the bamboo sticks. So, these three kind of materials was used as the packing material in this method: the beechwood shavings or the corn cobs or the bamboo sticks. So, this unit was embedded with one of these materials, and a bacterial film used to develop on the alcoholic broth, and the alcohol which was intermittently transferred into the tank, that is, it was irregularly transferred to the tank and the proper aeration was through the air inlets at the bottom of the tank. So, as the alcoholic broth which was fed into the tank, so this is the feed line from where alcoholic broth was, it is fed into the tank here, and these beechwood shavings, they are inoculated with certain Acetobacter species, that is the flora of Acetobacter species. So, as the alcohol trickled down through these beech shavings which were inoculated by Acetobacter species, a layer of the bacterial film used to set on these beechwood shavings material, packing material. And as the alcohol used to trickle down the beechwood shavings, they used to get oxidized, the alcohol used to get oxidized into the acetic acid. And these alcoholic broth, this was recycled for many period, for different cycles, for the better production of the vinegar. And in the bottom, this is the collection chamber, where all the filtered out vinegar used to get collected. And this is the tap outlet, and here temperature knob was used to provided in order to control the temperature. The optimum temperature in this process was 15 to 34 degrees Celsius, which is the optimum temperature required for the growth of Acetobacter species, and this temperature is generally inhibits the growth of certain other bacteria other than the Acetobacter species. And this method was very fast method firstly, and it used to produce the 15% of acetic acid. So by this method the results were quite good. Secondly, this method was used mainly, it is used for the production of white vinegar, which is generally used as a table vinegar also. Now the biochemical reaction that occurs during the fermentation of vinegar, in this process, vinegar is produced along with the water. Now there are different varieties of vinegar that have been produced since the last few decades. For example, cider vinegar, it is produced from apple juice. Now different varieties of vinegar are, firstly the apple vinegar or the cider vinegar, which is produced from apple juice. Secondly, cane vinegar. Cane vinegar is produced from sugar cane juice. Then wine vinegar is produced from different kind of wine. Then beer vinegar. Kiwi fruit vinegar is also produced, and it is developed in China. Kiwi fruit vinegar is generally developed from the kiwi fruit waste. Then rice vinegar, then white distilled vinegar, which is produced from distilled water or the distilled spirit. And some other kinds are date which are produced from the dates. Some are produced from barley. So these are different kind of varieties of vinegar. Now last one is the application of vinegar. Vinegar it has got different applications, like in food industry. In case of food industry, it is used in picklings. Then it is used as food condiment. Now white vinegar or the distilled vinegar, it is generally used for household chores. That is for this, it acts as a disinfectant, and it is used for cleaning glasswares in the house and also for cleaning the floors. Now some kind of vinegar, they have got medical uses also. For example, they act as antibacterials, and they are used in wound healing. Then they are also used in blood sugar level control, and it is also used for the treatment of diabetic patients. And it has also been found important in cervical cancer screening. So one of the major uses in medical is the cervical cancer screening. Now in agriculture, it is used as a herbicide. Now vinegar, it is the white vinegar or the distilled spirit vinegar, it is generally used in household chore because it is quite less acidic, that is, it is only 5% acidic. Thank you.